Not yet published results. Okay, great. Thanks for. Okay, great. I'm going to share my screen now. Good. So everybody should be seeing my screen right now. Great. Okay. So my PhD, I'm doing it at the University of Montreal in Quebec. So I'm speaking French, so sorry if my English is not that well. And I'm supervised by Étienne Lacroix-Carignan, a new professor at University of Montreal, and by Julian Starr at University of Ottawa, as well as Robert F. Nagsy at the New York Botanical Garden. And I have the financial support of many organisms, including the FBO. So thanks again for financing my work. So I don't know exactly the level of knowledge of people about Carex, so I'm going to do a quick wrap up about what's a Carex. So Carex are monocots in the Saperaceae family, and they are extremely diverse. It's by far the most diverse genus of plant in Canada in terms of number of species, and in Ontario as well. It's also the fourth biggest plant genus worldwide. There are more than 2,000 species, mostly distributed in Eastern North America as well as Eastern Asia. And there are Carex for almost every habitat you could think of, especially in temperate areas. So we have Carex in mountains in Africa. Carex cognata is one example, found in the Drakensberg region of South Africa. There are species up high in the high Arctic, like Carex saxatilis, Carex membranacea. Other species grow on very dry soils, like rocky ledges. Other in the sand, like Carex pumila, that grows in unstabilized dunes. And most species, I would say, are found in wetlands, where some are very keystone species and are important components of temperate wetlands, like Utriculata and Mesocarpia, as well as many others. Just to make sure everybody know the terms, I'm gonna explain them a little bit. So a bit of Carex anatomy 101. So the periginium, periginia is the plural of periginium. It's a structure that you see, it can take many colors. It's usually green, but in Carex aurea, for example, it's yellow. And it's a foliaceous bracts that envelopes the fruit that's enclosed inside. It's called the achene. And the achene it comes in different shapes, and it's very useful to differentiate species, especially in my group of interest, the section Lupulinae. And all we can tell apart the different species of Carex, it's mostly based on the analysis of these structures, the periginia, the achenes, and also the pistillate scales in some cases. And the species in my section, it's a relatively small section, only 6 species worldwide. And they're unique by the sheer size of their periginia. They have the biggest periginia of the whole genus Carex. And those 6 species, they're only found in Eastern North America in wetlands, mostly swampy areas. These species, here they are. I'm going to present them real quick. So there's the hop sedge, Carex lupulina. It's one of the commonest species in eastern swamps, mostly in rich woods. We don't see it much in the boreal forest. There's Carex lupuliformis that's quite widespread from Ontario and Quebec south to Florida and Texas. But even though it's widespread, it's rare pretty much everywhere. So it's a very interesting species, and it's considered threatened in Canada. Carex gigantea, on the other hand, it doesn't grow in Canada. It's only found in the southeastern United States coastal plain. So from about the state, it was once present in Pennsylvania, but it's extinct now, and it's found south to Florida and Texas. And the other species in the middle, Carex louisianica, is also not found in Canada, although it's found in the Cleveland area, very close on the south shore of Lake Erie. But it's also a species that's restricted to the southeastern United States coastal plain. The 2 other species, Carex intumescens and Carex griei, are found in Ontario and Quebec almost everywhere, but unlike all the other species, they're not really in the same group. What I say is that they're in the section Lupulinae, but what researchers found, and I mean, most of them expected this, is that the section Lupulinae is not natural at all. In fact, those 2 species, intumescens and griei, are only very distantly related from all the rest. Some people have elevated these into different subsection of the section Lupulinae, the subsection intumescentes that comprises these 2. And I'm not going to talk much about these in my presentation. I'm mostly going to focus on the subsection Lupulinae which includes Caractlusenica, as well as these 3 other species. So this subsection Lupulinae, is it a well-understood group? Well, looking at the literature, the answer will be a resounding yes. These sedges are all pretty big species as well as sedges are concerned, and there is a consensus over the status of all the species recognized in all recent taxonomic works. In fact, all the currently recognized taxa have not changed in the 1860s, which is quite remarkable for the genus Carex, because there are so many species described there all the time. And I've come to this, where do these undescribed species hide? When I was young, I used to think that, oh, all the things that are left to discover, they're mostly in tropical areas and very remote places. And yes, that is true to some extent. But for the genus Carex, for example, on average, there's 1 new species described every year in North America alone. And that statistics has held for 20 years. So it's very interesting. And those places in North America are not even that remote. For example, yeah, Disney World, it's actually a place where Carex are found. In between Disney World, in between Dino World, sorry, and the place where there is snow in the middle of summer, there's a nice wetland complex. And in that wetland, there are sedges, and not any kind of sedges, actually a species new for science that I'm about to describe. But yeah, I mean, more realistically, the best place to look for if you're searching for new species is not to go in Disney World, although it's almost as fun, is to go visit herbaria. And herbaria are very invaluable for me and pretty much all taxonomists out there, because they offer huge opportunities to look at thousands of species and specimens from all over the world in all different centuries. And they can really help in spotting the specimens that look weird or appear unusual or have weird distributions. Because, I mean, I'm a student and I can't afford to travel everywhere in the world, although I would love to. And one of those specimens was quite serendipitously sequenced as part of a barcoding project led by Julian Starr in the early 2010s. And by sequencing this herbarium specimen, this one actually, they found that it had unique mutation in its DNA that they found in no other species that sequenced so far. And while looking in herbaria at other weird specimens that shared the same peculiar morphology, they found another one from Georgia. And by sequencing this other specimen, they realized that, oh, well, they share exactly the same weird mutation. What's going on here? And so they thought, okay, maybe we stumble upon a new undescribed species. What can we do about it? And well, as it often happens in taxonomy, those specimens stayed on the herbarium shelf for 10 years or so until a student like me came over and decided to work on this issue and make sure that, okay, is it really a new species? And if so, give evidence to provide that support. And so that was the first hypothesis of my master's project, to see if these weird individuals could be distinguished and if they had a unique barcode and a unique morphology. And all these different evidence will support the recognition of this species as distinct, and I would need to find a name for it, basically. And we did fieldwork in Florida. Here on the top left, you can see my supervisor, Étienne Lacroix-Carignan, as well as Rob Nagsy. Rob is the tall one. He's so tall. And we visited a lot of beautiful swampy areas in Florida and pretty much all over the eastern part of the United States. And we collected quite a lot of specimens in my group, more than, I think, 300 specimens. We have a lot of specimens on loan if you want them. And so first, I wanted to test if this potential new species was genetically distinct. So to do this, first I extracted DNA from the Carex vouchers, both the ones that I collected in the field and herbarium specimens. And after that, I amplified with PCR some specific barcode markers that were proved to be informative to tell species apart. These were developed by Julian Starr and other people. One of these is the marker ETS. They mostly have weird names like these, so don't be afraid too much to look at these. Some of these barcodes come from the nuclear genome, and other barcodes come from the plastic genomes. That's of some importance, and I will explain that later. So by sequencing these, that's the output that we get from the sequencing machine. It provides what we call a base call. Basically, it tells, oh, at this place, there is an A base, adenosine, And the little curves that you see is the strength of the signal that's produced by the machine. And the higher the peak is, well, the more confident you are that this base call is good. In some cases, those base calls can overlap because, for example, for diploid species, we actually have 2 copies of the whole genome inside a single nuclei. And well, those 2 copies of the genome can differ to some extent. And when one copy has a base and another copy has another base, well, those those 2 base called peaks will superpose over each other, and we will see some interesting patterns that can be useful to recognize hybrids, for example. And after we get those sequences, we can align these from a bunch of different specimens that we can get and see the relationships between them to see if some specimens have sequences that are more close to each other. And if another weird specimen has a lot of different mutations that are not found anywhere, it will appear more distantly related if we do some phylogenetic analysis on these. And that's what I did, basically, a phylogeny of the Quebec subsection Lupulinae. So here we can see the compressed result. If you look closer, you can see that I did not show all the specimens that were included in that phylogeny, especially for the potential new species. You can say I sequenced 15 different specimens from pretty much all the United States from the eastern part. And all of those specimens had basically exactly the same weird mutations and were genetically very close to each other, and were distinct and distant from all the other species in the subsection Lupulinae. Carex gigantea was recovered at the basal species in the whole clade, and the 3 other species currently recognized in the subsection Lupulinae are also well recovered in the phylogeny. The clade support is not that high. I will address that in the future of my thesis. But what's important here is that the clade themselves are very strongly supported, even though the relationship between those clades is not so supported. So yeah, genetically, this potential new species is pretty strongly supported in that phylogeny. So that's good. So the second objective, well, okay, it's genetically distinct, but can we actually tell it apart in the field from other related species? Because, we don't carry a sequencing machine when we go out there in the woods. And to do this, I measured a bunch of herbarium specimens, more than 300 actually. And I measured more than 64 characters and a bunch more that I measured and extrapolated from these. And in the end, my dataset was quite complete, I would say. And I measured pretty much all the characters and the variables that thought at the beginning could happen distinguishing the different species in the group. And to see if I could distinguish morphological clusters in that data, and each cluster will ideally anyway correspond to a distinct species, I did what is called a discriminant analysis. So how does this work? A discriminant analysis basically is an algorithm. It's an algorithm that maximizes the within class variation and maximizes the inter-class variation of the data. But these 2 distinct parameters are kind of a compromise. And first, we have to calculate the first discriminant axis, and we do this as shown here on the figure. And once that's done, and we find the first discriminant axis, we can do the same thing for another axis that's perfectly perpendicular to the first, and find another axis that can explain the remainder of the variation in the data. And I trained this model using at first only DNA validated samples, the samples that I had sequenced, and I was sure that, well, they belong to the species that I thought they belonged to. And so here they are on the graph. We can see that these different samples group fairly closely well together. And when we added and tested the models using the samples that I had not DNA validated, well, we can see that, again, they cluster up pretty well. And that's only the first 2 discriminant axes, okay? If we look at, let's say, the first 3 discriminant axes, so in 3D, we can see an even better separation of the different clusters. And for the new species that we can see here in yellow is perfectly separated from Carex gigantea. That is the species that's morphologically the closest. I also did a classificatory discriminant analysis to see if, by just training the dataset and testing it, how many misclassifications there were. And there were almost none, basically. The model was extremely accurate in telling apart the different species. That model was used, it's called supervised discriminant analysis, because to train the model, I tell, okay, this sample that's been validated, it belongs to this species. But there are other algorithms that are unsupervised. It's basically you throw all the data at them without telling them, oh, this sample belongs to this species. You let the algorithm create the clusters by itself and see what kind of clusters it arrives up with. And I use Gaussian mixed models to do this. I'm not going to go into much detail, but basically the points that you can see on the graph on the left, the higher they are, the better the model is. And all those weird letters that you can see are all types of different models with different parameters. And the models with 5 clusters, that is 5 species, show up as one of the best compared to the ones with only 4 currently recognized species. That's what's currently happening in the subsection Lupulinae. The cluster that you arrive to correspond fairly well to species that are currently recognized. In fact, well, if you can see cluster name, they correspond fairly well to, well, recognized species. So that's great. And for the models that only admit 5 clusters, that is the 4 currently recognized species in subsection Lupulinae plus the putative new species, well, the model is more than 98% accurate. So morphologically, there is no doubt that this new species can be distinguished. And not only with those models, you know, with characters that can be assessed in the field, like achene shape. This new species has achenes that are more wide toward the middle, whereas in Carex gigantea, it's wider toward the top of the achene body. And there are other slight differences too in the shape of the perigenia and other characters like these. So we can see there is very little overlap between this new species of Carex and the other species for those characters. And for example, this scatter plot also shows it pretty well, that the separation is nearly perfect. And those characters can be measured by botanists in the field. So that's reassuring for some of you, I suppose. And when I was in the field, I also collected live samples that I brought back to Canada to be grown in a greenhouse side by side to see if the plants that were grown together showed different phenotypes. And yes, they did. Even though they were grown in exactly the same conditions, they flowered at different periods and had very different spike morphology, if we can see on the left. So that's, again, another strong argument that these two species are distinct, and that morphological differences is not only caused by environmental differences or differences in water level where they grow. And the distribution of this new species, it's actually fairly widespread in Florida, and it goes up north to North Carolina. But the farther north you go, the rarer it gets, basically. Around the Tampa Bay and around Orlando, where Disney World is, the species is actually fairly common. Where I collected the type specimen at the Withlacoochee State Park, there is actually a stand, an uninterrupted stand of this species that of millions of individuals over like 2 or 3 kilometers long along the river. So it's not by any means a rare species. It just evaded detection for quite a long time. And the name I chose, it was Carex gator, because it grows in gator habitat, basically, and in those swampy woods with bald cypress. And yeah, when I collected the species, I didn't see any gator, unfortunately, or fortunately. I would have loved to see some of them, but from far away, you know. But yeah, it took me a while to find the name, so I hope you find it good. And so that wraps up the first part of my project. At first, it was supposed to be a master's thesis, so that was supposed to be the end of my project. But as it often happens in science, we stumble upon unexpected things. And so my master's thesis turned into a PhD thesis. And again, I'm going to start with this. Lupulinae section, is it a well-understood group? Well, I gotta prove that, yes, we were wrong. It's not as well understood as we once thought. 3 main problems arose when I searched for literature is that, well, we don't really know what Carex lupuliformis really is. And again, we'll come to this later. Also, many people suspected that hybridization was frequent within the subsection, but no one really dove deeper into it. So I also wanted to answer this question, like, is hybridization frequent among the species of subsection Lupulinae? And third, are there some other cryptic entities involved, not only Carex gator, but maybe some other pesky things hiding somewhere else? And yeah, down the rabbit hole we go. And I'm in the third year of my PhD thesis, and I'm probably halfway. So there's still a lot of work to be done. I'm going to start with the strange case of the hop sedge and the false hop sedge. First, I'm going to present again these two species. The Carex lupulina, the hop sedge, it's the common species that's found pretty much everywhere in Ontario and in southern Quebec. And the Carex lupuliformis, it's rare in southern Ontario, and Will did some surveys to find a few remaining populations. We can see the differences between these two species rely mostly on the shape of their achenes. For Carex lupulina, the achene shape is mostly longer than wide, and they don't have like those thick knobs on the corners. For Lupuliformis, these knobs are, the shapes differ between different samples, but they're mostly prominent. And there are also differences in the perigynia arrangement. They are mostly laxly arranged in the spikes for Lupuliformis and are more densely arranged in Carex lupulina. And also, Carex lupuliformis, at least in Ontario, is a strictly rhizomatous species with leaves that are a brighter shade of green compared to Carex lupulina, that is mostly a cespitous or shortly rhizomatous species with darker green leaves. But we kind of find intermediates between these two, especially in Ontario, and Will could probably tell a lot about these. I call those colloquially like Carex lupuliform-ish, because they were kind of intermediate between two. Those forms are especially very common actually in Florida and in South Carolina. There are some of these weird intermediates in Ontario as well. And so I wanted to know, okay, what the hell are these? And are they really Lupulina, Lupuliformis, or are they hybrid between the two? So to assess this and to assess whether there is introgression between Carex lupulina and Carex lupuliformis, introgression is basically like hybridization that happens during a long period. Basically, hybrids, we hybridize with either of their parents, and there's a lot of gene flow between two species. Using the method I use for Carex gator, it's not really the best method, because those markers usually have a fairly low number of variable nucleotide sites. And the method I'm going to use, sorry for this, is using RAD sequencing. That generates a lot more data for a lot more loci in the DNA. And so it's a much more robust and reliable method to detect if there is introgression signals in the genomes. And I sequenced a bunch of Carex lupuliformis and lupulina specimens, and it came as a surprise that, well, they do not form a monophyletic group. Actually, Carex lupuliform-ish is quite, it's spread along the phylogeny in a bunch of different places. The first group that we can see at the top of the figure, these are specimens from southern Quebec and northern Vermont. And these Carex lupuliformis samples, they have achenes that are much longer than wides and have like very thick knobs on the corners that are bulging, you know. And these plants are cespitous, and they grow along rivers in silver maple swamps in areas where there is a lot of water level fluctuation, in rivers that are usually not dammed. And so that's the habitat of the species. I think there are some specimens of these that were collected in Ontario. One that comes to mind is one that was collected by Mike Oldham in Essex County. I think the population disappeared, so I couldn't visit it, unfortunately. And there might be other, like the specimen from the Galt region. It's not clear if it was really collected in the Galt region, but anyway, it may also belong to this entity. But the one that's most commonly found, well, I say commonly, it's still very rare in Ontario, is the other form that's called here Carex egartii. That's the thing that's found in Ontario in vernal pools, in very rich situation, in mostly vernal pools that are not connected to any hydrological feature, I would say. It's not along big rivers. It's little pools in the middle of the woods that are full of water in the spring, and they dry up slowly in the middle of summer until the fall, where they are almost dried up but not quite. And this Carex lupuliformis that's in Ontario is the rhizomatous one. And the lupuliformis and lupulina, well, they grew kind of closely together. And the difference between these two forms of Carex lupuliformis that I found using DNA, Joyce Hoffman, that did her thesis on exactly the same group of Carex that I did in 1974, also found the same difference. And back in her days, DNA wasn't even a thing, so she saw those differences based purely on morphology, and she also created a small distribution map. Basically, the Carex that I call Egertii, the one that's found in southern Ontario, is the species that's the most common of the two, and it's quite widespread in the southern United States, in southern New England, like in Connecticut, and also in southern Ontario. Whereas the other form, that's the thick knobby one, the one that is found in Quebec, for example, this is much more restricted. For now, the only sequence samples that I have, they come from Vermont, from Quebec, and also from Illinois in the Chicago region. Basically in the same spot that she wrote on the map that she created in 1974. So I still have to sequence more specimens to really define better the distribution of that entity, but it appears, at least for now, to be morphologically distinct and also genetically distinct. So the next step of my project is to basically do rad-seq analysis and to sequence and sample more individuals of this rare sedge. That's basically why I was financed by the FBO, to search for the extant populations of Carex lupuliformis in Ontario, to try to determine whether they're one or the other of these two entities. I found that this Carex lupuliformis was grouping much more closely to Carex lupulina than it was to Carex lupuliformis, both genetically and morphologically. And for now, I consider lupuliformis to be just a broad-acting form of Carex lupulina. And they share basically the same habitat, same ecology. It's just some random individuals that happen to have these broad-actings. I don't exactly know why that happened. Maybe like some mutations in the DNA. I don't know at this point. So yeah, these are the two clades of lupuliformis. They are genetically, very distinct. And so that begs the question, what name is applicable to Ontario's Carex lupuliformis specimens if there are two species?
It depends on where.
Well, I think somebody has problems with their mic. Could somebody mute them, please? Because I don't know how to do this when I'm presenting. Thank you. And so to make sure what really is Carex lupuliformis, we have to look at the type specimen. The type specimen of Carex lupuliformis is actually under the name Carex lupulina variety polystachya. And when it was elevated at the species rank, well, it couldn't name it Carex polystachya because the name polystachya already existed. So they chose another name, Carex lupuliformis. But the basionym, the name on which Carex lupuliformis was based, is Carex lupulina variety polystachya. And the type specimen at the Harvard Herbaria looks like this. These are images of the type. And as we can see, the achene are not very good looking, if I can say that way. These achenes are all parasitized or immature, aborted. None of them are well developed and show these characteristic achenes and those knobs. So by looking at this specimen, it's very hard to say whether they belong to this achene shape or the other, because it's just a mess. So, and I couldn't, unfortunately, sequence this type specimen. So I had to look at the type locality. That specimen was collected in the state of New York, in Phillipstown. So that's basically, how can I describe it? Along the eastern part, close to the Connecticut border, and not along the Hudson, but more in the highlands, in vernal pools. Another name that could be applied to this thing is Carex egertii, that was described from Missouri, and that's that in shape looks a bit like Carex luisianica. I mean, the Perigenia that are more inflated resemble quite a lot the species, but the achein shape is fairly different. And this Carex agoutii could be a name that could apply to the specimens that are found in southern Ontario. I could sequence the type of agoutii, but as I cannot compare it with the type of the other specimen, I can tell for now to which of these entities the Ontario specimen belong to. Yeah, about the type specimen collected in New York. I went with Patty Butter at the type locality in Phillipstown, and I collected specimens, fresh specimens that I could sequence. And they're not sequenced yet, so I cannot give you any more results, but I will be able at least to say to which genetical cluster the type specimen most likely belongs to. And also, that I didn't really answer the question, are there hybrids involving Lupuliformis? Because I suspect there might be, but they're much more rare that we won't believe they were. This one is from the Quebec. On the left, you see a specimen of Carex lupuliformis. In the middle, the putative hybrid, and on the right, Carex lupulina. But most of these hybrids that are found in herbaria are mostly not really hybrids. They're mostly morphological abnormalities caused by parasites. Because believe it or not, there are a lot of insects that feed on these sedges, on the acheins, on the perigenia, and on the roots and on the leaves. And here we can see abnormal acheins that are deformed by a larvae of a small dipterian species in the tribe Dasinorini. That was sequenced actually, and is probably a new genus because nobody ever sequenced those small flies. And that's those weird achein shape. Some people in the past believed that they belong to a new species, but they're only really deformed acheins caused by the small fly. Another weird morphological abnormalities that can be caused by parasite are these very narrow and slender perigenium that are barely inflated. And those are most likely caused by an aphid, Carolinea caricis. Those are the ones that I sequenced. And yeah, that's again a very elusive aphid that's very poorly known. And it feeds on the sap that's on the perigenia. As we can see the cells on the perigenia membrane actually on the far right. And those little tiny bugs feed on these, and the perigenia, while they develop, they take on unusual shapes. And again, those weird morphological abnormalities were assigned a name in the past, Carex bellavilae, that you can see on the right here. That's the type specimen. And yet another species, they don't feed on the achein, they feed on the perigenia. So another fly, you can see the larvae on the far left. And these little wasps, when they put their eggs inside the perigenium, they poke little holes in them. And we can see these on herbarium specimens. It's pointed by the arrow there, the small blackish dot. And those perigenium that are parasitized by this species of fly, they take unusual shape and they kind of inflate a little bit, and they give unusual aspect to the spikes. And again, these were considered by some to be hybrids, but they're not really the case. So are Carex hybrids even real? Well, I can say that yes, they are. For some hybrids that I found, they are, it's impossible to mistake them for anything else. This hybrid was collected in southern Carolina, and it's a almost fully sterile hybrid between Carex lurida and Carex lupulina. And it has intermediate morphology, and it grew right in between the two parents. And when I sequenced these, looking at the sequences, you can check that it's intermediate in the sequences between the two parents. And when I talked before about these peaks that were overlapping in the sequencing machine's output, well, that's how I could detect these putative hybrids, because one copy of the genome belonged to Carex lurida and the other copy of the genome belonged to lupulina. And so these overlapped in some places, and I could detect these. Another hybrid that was found quite recently in Ontario, this one, was Carex lupulina hybridized with Carex retrosa. Again, the hybrid grew right in between the two parents and was again fully sterile. And again, genetically, it was perfectly intermediate between the two parents. This hybrid is not very common in Arboria, but it's likely underreported. I collected this one with Michael Oldham in the Kawartha Lake region, I think. No, it was around the Belleville, sorry. But yeah, and it's one to look for. And I collected it again in the Ottawa region along the Kanata area. So as long as Carex lupulina and Retrosa grow together, this hybrid could be found. And when they grow, it's quite vigorous. The clumps are big and it shows hybrid vigor. And it's quite unmistakable when you see it in the field. And just to make sure, you just press the spikes and see if there are any achenes, any bumps that you can feel. And if it's the hybrid, all the perigynia should be empty. And well, I also unearthed some old taxonomical problems while doing my thesis and searching the literature. One of these problems was Coryndocarex macunii. So that species was described in 1866 by Dewey, and Kenneth Mackenzie in his latest taxonomical treatment in 1920 something, he basically wrote this, that all of these names that are currently mostly unresolved, he grouped them all under the name Macunii and said, well, this name covers a series of hybrids in which more than two species are concerned, but I cannot tell what the hell the parents are. So that's the situation, and nobody else tackled these problems until now. So these names, if you look at them in most databases, they're put mostly under Carex lupulina. Some of them, they are hybrid, but between other species not even closely related, like Carex ateroides. So all these names are problematic right now, and I wanted to solve this issue. And I think I mostly succeeded. So this is the type specimen of Carex macunii that was collected by John McCown in Ontario in Seymour County, so in the Belleville area. And those spikes are very lax, and they look somewhat like those of Carex lupuliformis. And that's why some people believe that Carex macunii was actually a sterile hybrid between Carex lupulina and Carex lupuliformis. But as some of you know, Carex lupuliformis is found nowhere near close this locality, so that's very suspicious, if I can say. And I collected this summer, actually, a specimen in Quebec, right along the Ontario border near Fort Coulonge. It's about 45 minutes west of Ottawa. And the specimen I collected, it fits extremely well with the type specimen of Carex macunii. And this specimen was actually not a hybrid. It was a whole population with only these weird aberrant forms. And population means specimens over 100 and 200 meters. So that's definitely caused by some genetic mutations. But to tell whether if there are new species or just some abnormalities, I have not yet sequenced that specimen, so I'm really eager to know what that is, because those achenes were all fully fertile and barely distinguishable from Carex lupulina, and it grew right alongside more typical Carex lupulina. So at this stage, I really don't know what to make of this, but all I can say is that this is not a hybrid because it's fully fertile and self-perpetuating. So yeah, first problem that I'm trying to solve. The second one, that's other photos because I wanted to show you more pictures of this very weird sedge. The spikes look almost like Carex gigantea. It's very hard, and that's the habitat. So it's mostly vernal pools with cedar and hemlock. It's not a very special place, really. So it was quite a surprise when I found these. And actually found these because someone else on iNaturalist posted pictures of weird Carex lupulina, and I just thought, okay, these definitely look weird. And so I had to go there to find them myself. Yeah, another problem, another species, Carex bellavilli. This one was also collected by John McCown, again in the 1860s, and it was never collected ever since. Species was described by Dewey, and it's a very unusual specimen, very robust, and the achenes are all not aborted, but they are parasitized, as we can see on the left. This achene had a fly larvae inside of it, and it was completely empty. And those perigenium were, again, very not inflated, weird. And well, you probably remember the picture with me and those aphids. That's the good candidate that we collected in the Belleville area. At first, I thought that, well, okay, they were full of aphids. Okay, it's probably just caused by weird parasitisms. And just to make sure of that, I collected a part of this clump and I grew it in the Montreal Botanical Garden greenhouses. And I waited for them to flower and see, okay, well, I know there are no aphids right now in the greenhouse, so how will the flowers and the spikes turn out? And if they turn out as typical Carex lupulina, well, then we will have our answer. And if it shows up as some weird things, then well, it showed up as some weird things. So basically, it looked like this. Yes, so that doesn't look like a normal Carex lupulina. And I sequenced that specimen, and indeed, it is not closely related to Carex lupulina. And it showed up as actually it's a hybrid between Carex lupulina and something else. That something else currently does not have a name, and I'm serious. So, and I collected this other thing that grew right alongside this hybrid, and I have a really ugly specimen, so I want to search for more specimen of this weird thing. And here are some image of the specimen that I sequenced, and you can see genetically, it's just very unusual. It shows up close to Carex retrosa and Gigantea, but it's not the same as the hybrid between Lupulina and retrosa that I showed you earlier. It's very different. And the achenes of this are fully fertile. And so, yeah, I really don't know what to make out of this. So it's probably an elusive undescribed species. And here are the achene shapes of all the three. So Carex lupulina on the left, retrosa on the right, and right in the middle, this putative new species with really thick and short achenes. Right now, it's a mystery to me. So really, I want to ask your help to find more specimens of this really weird things, because it's found in the Belleville and it's not in any special place. That's from the H.R. Frink Conservation Area. And there were other specimens collected in around the Kawartha Lakes that could belong to this undescribed entity. And another one that got sent to me from the Chicago region that might be, but I've not sequenced it yet, so I really don't know what to make out of this. But yeah, I sequenced it, and it's different. And the hybrid that's growing in the greenhouse right now also shows, yeah, it's so weird. Even now, I don't really believe it, but still, an interesting fact, yes, the only extinct plant species that was endemic to Canada was also happened to be found in the Belleville region too. It's a moss that was put in the genus Forstremia, and so maybe the Belleville region is an endemism center. It's so interesting and weird. So I really want to collect more specimens of this elusive entity because yeah, it's definitely a new species. Yeah, so right now I found it in those areas. I've only sequenced one specimen, so I'm still waiting for more sequences, and I'm not entirely sure if all of these actually belong to this new entity. But yeah, so in case it helps you with in your searches of this Carex. And so basically what you should be looking for is specimens that look intermediate a little bit between Carex lupulina and retrosa, but with fully developed achenes that are really not that long and quite thick. And the perigenium are usually smaller than what's typical for Carex lupulina. Yeah, that's some other weird specimens from the Kawartha Lakes region that I found in herbaria, again with fully fertile achenes. So maybe the only weird forms of lupulina, but maybe there's something else. So I can't wait to sequence these. And the next step is to build phylogeny using RADseq, and I have some preliminary results for you. So basically, that's the Lupuliformis from Pennsylvania. That's different from Carex lupuliformis from Quebec. That's the new species of Carex gator. And that's the putative new species from Belleville. So these three things that I put in red are new things that we didn't think would exist in a section that we thought we understood well. So basically, yeah, I'm going to explode the diversity in this group by describing probably 3 more species or resurrecting them during my PhD. So yeah, that's a really big surprise. So I'm really happy that I didn't stop at the master's thesis and I dove all the way to the PhD. And so that ends my presentation. And if you have any question, tag me on iNaturalist. I will be very happy to get some nice pictures of those carex. And if you can send me specimens, that's even better. I could sequence them and make sure they really are the new species. And so, yeah, if you have any question, feel free to ask them. And thank you all.
Thank you so much, Etienne. That was so cool. that was way cooler than I could have expected. There's a few questions in the chat. I'd like to ask anybody else who has questions, please put them in the chat. It's just easiest that way. First question I see is from Sam. He asks, I'm curious about the actual distribution of Carex lupuliformis. Is the lupuliformis in the southeastern US actually lupuliform-ish?
That's a good question. I don't have a distribution map to show you, but what I can say is that the thing that's found in Ontario is the thing that's most widespread in the southeastern United States. But, and there is a but, that thing is not found in Florida. And if you look currently on iNaturalist at the Carex lupuliformis distribution map, you will see that the highest concentration of dots and occurrences is currently found in Florida. And currently all the specimens that are from Florida that are identified as Carex lupuliformis, they actually all are Carex lupuliformis, and so they are all Carex lupulina basically. And I sequenced, I think, 10 specimens of Carex lupuliformis from Florida, and they all showed up nested within the Carex lupulina clade. So yeah, it's just a broad achene form of lupulina that people mistook for Carex lupuliformis. And I mean, people until now, they still don't really understand what is lupuliformis. So I really want to make sure people use a good key to distinguish these, because the achene, the thickening on the knobs of the achene is not the only characters, and some people focus too much on them. If that answers your question.
There's a question from Eric Ungberg.
Yeah. Thanks,
Wonder
Eric.
if the range for Carex aggertii that you showed is more or less complete, or did you find it elsewhere in your explorations? And then he has a second question about your thoughts on Carex intumescens var fernaldii.
Let me go back to the distribution map. Okay. So the map for Carex aggertii there is actually incomplete. I mean, I can, yeah, I will try to draw something using my cursor. If I can, yes, this. You should be able to see a drawing. So that's right here is currently the distribution of aggertii. So it's quite widespread. Do you see my red ugly thing? So it's quite widespread. I would say the place where it's the most common, and it's common nowhere. It's in Texas and Illinois. Like in those regions, it's fairly common, especially around the Houston area and in southern Illinois. There are also quite a nice concentration here in New England and along the Washington area, but there are still some disjunct occurrences in North Carolina and South Carolina. And I collected specimens from there that I sequenced, so I can be sure that these are really this aggertii thing. So does that answer your question regarding lupuliformis? I would say, yeah, he cannot answer. So yeah, regarding Carex fernaldii, Carex intumescens var fernaldii, I cannot answer your question fully, But I know that Julian Starr and University of Ottawa has this as a side project, and he wanted to work more on these. He has some unpublished genetic sequences of this species. So if you want, I would encourage you to contact Julian Starr at University of Ottawa. He could answer your questions. And I don't know if you want me to, spoil the fun for you.
Here's a final question from Quentin Wiersma. He asks, would Carex retrosa be better nested within section Lupulini?
That's actually a very good question. Right now, I don't have a definitive answer, but because there are some outgroups from Asia that I've not yet sequenced, and I believe they could group close to Carex retrosa. So whether Carex retrosa merits its own section or it's nested within Lupulina, it's still up to debate. But there are still some strong differences between Carex retrosa and Lupulina, especially in the shape of the achene and also in micromorphology. The achenes of Carex retrosa, when you look at them at very high magnification, the achene surface is covered in small silica bodies that form like papillae, bulbs on the center of the cells. And these are completely absent in all the species in section Lupulinae, subsection Lupulinae, sorry, because intumescence and Greer also have them, but they're distantly related to the rest. And yeah, Retrosa, it's still grouping close to the subsection Lupulinae. In the analysis of some of my markers, it actually was nested within the subsection Lupulinae, but overall, I feel like it may belong to a distinct section. But yeah, that's a very good question. I need to sequence species from Asia that might group closer to really answer it. But yeah, it's still a bit of an odd species for a subsection Lupulinae.
Okay, we'll take a final question here from Gavin. Just in the last 2 minutes, he asks if there's any possibility of Asian species showing up in North America.
For my group, subsection Lupulinae, no, because it's a strictly Eastern North American group. Apart from my PhD, actually, I focus on what's called the Hirta clade. It's a group of about 106 species that comprises species in the section Vesicariae, Paludose, Carex, and other things that are placed in more distantly related sections like the Squarose, but really phylogenetically they're closer to the section Lupulinae. And I can say that, well, there are some introduced species from the Hirta clade in North America, like Carex melanostachya, and I think Carex pumila was found somewhere. But yeah, from the species that are close to the subsection Lupulinae, no, and I've not seen any in Herbaria or in iNaturalist, and I've reviewed pretty much all the North American observation of these species, so I'm very confident when I say that. But it's not impossible if that someone plants them in a garden and it shows up as naturalized somewhere. It's still a possibility.
Awesome. Well, that was great, Étienne. Thank you for doing this for us. Those of you who are not members, if you're in Ontario, I really encourage you to go to our website and sign up for membership with the FBO. If you're not in Ontario, follow us on Facebook and Instagram. Yeah, happy holidays and see everybody later.